The room is dark, not the soft dim of a curtained afternoon, but the real dark of a night fully settled, the kind that has weight to it, that presses gently against the eyes and asks nothing. The body on the bed has been still for a few minutes now. The breath has slowed, the hands have opened, and somewhere in the brain, at the thin and wavering edge where attention begins to lose its hold, the first stage of sleep has quietly begun. It does not arrive with any announcement. There is no signal, no shift that can be felt from the outside. The eyelids have been closed. The muscles have softened. The sounds of the room, a car passing somewhere on a distant street, the low hum of something electrical, the faint percussion of a house settling, have begun to lose their edges. They are still present, but they have stopped carrying meaning. The border between the sleeper and the room is becoming permeable, and the brain is already somewhere else, moving slowly down through the first of 4 distinct territories it will visit before morning. That first territory is called N1. It is the lightest of the sleep stages, a place of transition rather than rest, a narrow hallway between the lit room of waking and the deeper chambers beyond. In N1, the brain's electrical activity begins to shift. The alpha waves that dominate a relaxed, closed-eye waking state, oscillating at roughly 8 to 12 cycles per second, start to give way to slower, lower amplitude theta waves, 4 to 8 cycles per second, diffuse and irregular, the signal of a mind releasing its grip on the world. The change is gradual. It happens over seconds or minutes. The sleeper may not even know it has happened at all, and if roused at this stage would almost certainly report that they had not yet fallen asleep. N1 is brief. In a full night's architecture, it accounts for only a few % of total sleep time. A soft vestibule, quickly passed through. The muscles continue to relax. There may be brief, involuntary contractions, a sudden twitch of the leg, a small jerk of the whole body, called hypnic jerks, the nervous system briefly and harmlessly misfiring as it hands over control from the waking motor circuits to the chemistry of sleep. The eyes move slowly behind the lids, rolling without purpose. The heartbeat has eased. The temperature of the body, very slightly, has begun to fall. From N1, the descent continues. The brain moves into N2, a considerably deeper and longer stage, and the electrical signature changes again. Here, among the background theta activity, 2 new waveforms appear. The first are sleep spindles, brief bursts of oscillation at 12 to 15 cycles per second, lasting only half a second or so, generated by the thalamus and sent radiating outward through the cortex in rhythmic pulses. The second are K-complexes, large, slow, biphasic waves, sharp and then long, that erupt from the cortex in response to sensory disturbance or appear spontaneously in the quiet of deep N2. Together, these 2 features are the hallmarks of N2 sleep, readable in the record of any polysomnographic study as unmistakable as a signature. They mark a brain that has fully committed to sleep, that is no longer passively drifting, but is actively maintaining its own unconscious state. No, too, is the most abundant stage in the night's architecture. Across a full 8 hours of sleep, it accounts for roughly half of all sleep time, returning in each cycle, its spindles and complexes repeating their quiet electrical language through the small hours while the body is still and the world outside continues. It is, in a sense, the reliable floor of the sleeping night, not the deepest sleep, not the most active, but the most consistently present, the background against which the other stages emerge. Then the descent reaches its lowest point. From N2, the brain enters N3, slow wave sleep, the deepest and most restorative stage in the cycle. The EEG record here is transformed. The theta waves and spindles give way to high-amplitude, low-frequency delta waves, oscillating at less than 2 cycles per second, vast and slow, and dominating the trace. These waves reflect the synchronized firing of enormous populations of cortical neurons, millions of cells rising and falling together in a slow rhythm, like a tide moving through the brain's outermost layer. The body is at its most deeply unconscious here. Arousal threshold is high. A sound that would wake the N2 sleeper barely registers. The muscles are relaxed, the heart rate and breathing have settled to their lowest rates of the night, and growth hormone is released into the bloodstream in its largest pulse of the day, beginning the work of cellular repair that slow-wave sleep makes possible. N3 is the stage from which it is hardest to wake. If pulled out of it by an alarm, by a hand on the shoulder, the person wakes disoriented, clouded, in a state called sleep inertia, the brain not yet able to clear the delta wave chemistry quickly enough to function normally. The deep, slow waves do not release the sleeper gently. They hold. The descent has now reached its floor, and then, without any clear signal, without the sleeper knowing, the brain begins to come back. Not all the way back to waking. Somewhere else entirely. The delta waves reduce. The brain climbs back through a kind of NREM reversal, the architecture briefly retracing the path it came down, and then it arrives somewhere that is neither deep sleep nor waking, somewhere with its own entirely distinct signature and its own entirely distinct character. It has arrived at REM. REM sleep, rapid eye movement sleep, was first identified in 1953 by Nathaniel Kleitman and his graduate student Eugene Aserinsky at the University of Chicago. Aserinsky had noticed that the eyes of sleeping infants moved in periodic bursts beneath the closed lids and wondered whether the same was true in adults. It was. When Kleitman connected his own son to an electroencephalograph and an eye movement recorder, the tracings that came back were unlike anything they had expected. The brain's electrical activity during these periods looked almost like waking, fast, low amplitude, mixed frequency. The eyes were moving rapidly in coordinated bursts, and yet the person was deeply, unambiguously asleep. The combination was so strange that Aserinsky initially doubted his equipment. He checked the recordings 3 times before accepting what they showed. What they had found was a separate state of consciousness, a fourth territory in the sleeping night, as distinct from slow-wave sleep as sleep is from waking. In the decades that followed, the techniques for measuring this state became more precise and more systematic. The study of sleep in a clinical and research context requires an approach called polysomnography, the simultaneous recording of multiple physiological signals from a sleeping person over a full night. A polysomnographic recording is a map of the sleeping body written in 3 primary languages at once. The first language is the EAG, the electroencephalogram, which records the electrical activity of the brain through electrodes placed across the scalp. In waking, these traces are fast and irregular. In N3, they are slow and mountainous. And in REM, they return to something fast and low and mixed, not identical to waking, but close enough that a person unfamiliar with sleep medicine, looking at the EEG trace alone, might mistake it for a recording of someone awake. The second language is the EMG, the electromyogram, which records muscle tone through electrodes placed under the chin. During NREM sleep, there is some residual tone in the muscles, a baseline electrical hum, the body maintaining a low level of postural readiness. During REM, this hum falls away almost entirely. The chin muscles go electrically silent. The skeletal muscles of the whole body reach their lowest tone of the night, a suppression so complete that it is one of the most reliable signals in the entire polysomnographic record. Something in the architecture of REM has shut down the body's ability to move in ways that are, in waking life, entirely automatic. The third language is the EOG, the electrocardiogram, which records the movement of the eyes through electrodes placed near the outer corners of each eye. It is this trace that gives REM its name. During these episodes, the eyes move rapidly beneath the closed lids, darting left and right and diagonally, sometimes in brief, organized bursts, sometimes in long rolling clusters, and these movements are not slow and rolling like the eye movements of N1 or the casual movements of quiet rest. They are quick, directed, purposeful in appearance, the eyes moving as though tracking something or looking for something in a space that exists only inside the sleeping brain. These three signals together, the low amplitude mixed EEG, the suppressed chin EMG, the bursting EOG, constitute the clinical definition of REM sleep. No single one is sufficient. It is their simultaneous presence that marks the state. A polysomnographic record of a full night lays these three traces in parallel bands, one above the other, and when all three shift into their REM configuration at the same moment, there is no ambiguity in the record. The brain has arrived somewhere specific, and the machinery of the body has changed accordingly. A full sleep cycle, the progression from N1 through N2 through N3 back through N2 and into REM, takes roughly 90 to 110 minutes in a healthy adult. Over the course of a typical night's sleep of 7 to 8 hours, this cycle repeats approximately 4 to 6 times. The body and brain complete their full revolution from the light edge of sleep to its deepest floor and up again into the strange bright territory of REM, and then begin again and again and again, each cycle a self-contained arc, each one slightly different from the one before. The differences between cycles are not random. They follow a clear and reliable pattern, a distribution across the night that says something deep about what the sleeping brain is doing and what it most needs. In the early cycles, the first and second of the night, slow-wave sleep dominates. 1-3 is long and deep, the delta waves abundant, the body pressing hard into the restorative depths of slow-wave sleep, as though paying down a biological debt accumulated through the waking day. REM in these early cycles is brief. It may last only 5 or 10 minutes, a narrow window at the top of the first cycle's arc, barely present before the brain descends again into NREM. By the middle of the night, the balance shifts, N3 shortens. The third and fourth cycles give less time to slow-wave sleep and more to N2 and REM. And by the final cycles of a full night, the fourth, fifth, sometimes a sixth, slow-wave sleep may be almost entirely absent, and REM has expanded to fill the space. A single REM episode in the fourth or fifth cycle of a full night can last 45 minutes, even approaching an hour. The brain, having completed its deepest restorative work in the early hours, now devotes the long approach to morning almost entirely to this other state, this bright, active, paralyzed, internally generated state that is as different from slow-wave sleep as anything in the body's biological repertoire. The result of this skewed distribution is that the proportion of REM sleep in a full night is concentrated almost entirely in its second half. A person who sleeps 7 or 8 hours and wakes on a natural schedule has experienced most of their REM sleep in the 90 minutes before they open their eyes. Cut the night short by 2 hours, an alarm set too early, a night ending before its natural conclusion, and the loss is not evenly distributed across all stages. It falls disproportionately on REM, which was still accumulating when the night was cut. Across a full night of undisturbed sleep, REM accounts for roughly 20 to 25% of total sleep time. In an 8-hour night, that is approximately 90 to 120 minutes of REM, distributed in episodes of increasing length. The first short and tentative, the last long and generous, all of them together forming an arc that is as regular and predictable as the architecture of the night itself. This regularity, this consistent nightly cycling, belongs to a broader category of biological rhythm called the ultradian rhythm. Unlike the circadian rhythm, which operates on a roughly 24-hour cycle and governs the timing of sleep and waking, the ultradian rhythm operates on a much shorter time scale. In the case of the sleep cycle, roughly 90 to 110 minutes per complete revolution. It is a faster clock, nested inside the longer circadian one, repeating 4 to 6 times within a single night. The two clocks do not compete. They nest one inside the other, the short cycle spinning within the long one, together producing the full architecture of the sleeping night. The ultradian rhythm of sleep is not unique to humans. It has been observed in various forms across a wide range of mammals. The duration of the cycle scales roughly with body size and metabolic rate. Smaller animals cycle faster, larger ones more slowly, but the basic architecture of NREM and REM alternating in sequence appears wherever sleep itself appears. It is an old arrangement. It predates anything that could be called a human brain by a very long time. In the human brain, the signal that drives this cycling does not originate in the cortex. It rises from older, deeper structures. The suprachiasmatic nucleus of the hypothalamus governs the circadian timing, the broad envelope of sleep and waking across the day. But the shorter ultradian cycling, the 90-minute rhythm within the night, is generated largely by the brainstem, the most ancient region of the brain, the part that evolved first, and that handles the most fundamental functions: heart rate and breathing, the regulation of arousal, the switching of sleep states. The brainstem does not need the cortex to run its clocks. It runs them in the dark without input, night after night, with a reliability that requires no conscious participation and no memory of the night before. The surface of the brain, the cortex, with its elaborate wrinkled architecture, its billions of neurons arranged in layers, its vast associative networks, rides on top of this machinery like weather riding on the curvature of the earth. The cortex produces the complexity, the memory, the reasoning, the language, the dreaming. But the basic rhythms that govern when sleep comes and how it cycles belong to much older regions, and they were working long before anything like a cortex existed. A person lying in a sleep laboratory tonight, connected to the sensors of a polysomnographic study, will complete roughly the same arc that every healthy adult completes every night, the same 4 stages in roughly the same sequence, the same ultradian cycling, the same gradual redistribution from slow-wave sleep in the early hours to REM in the late ones. The record of their night will look in its broad structure very similar to the records collected in sleep laboratories all over the world over the past 70 years. The individual details will differ, the exact timing of each stage, the amplitude of each spindle, the precise duration of each REM episode. These are as individual as a fingerprint, but the architecture beneath them is shared. The polysomnographic record of a full night is, in a sense, the closest thing science has to a complete portrait of a sleeping mind. The 3 parallel channels, EEG above, EMG in the middle, EOG below, each tracing a different aspect of the same body, running in parallel across the hours from lights out to morning, together capture something that the sleeper never directly experiences and never directly remembers. The sleeper is inside the record. The record is the outside view of what it means to sleep. On that record, the first appearance of REM can be precisely located. It occurs in a healthy adult without sleep deprivation or mood disorder, somewhere between 70 and 120 minutes after sleep onset, after the quiet crossing of N1, the first long descent through N2, the deep valley of N3, and the return climb back toward the surface. The elapsed time between the moment of sleep onset and the moment the record first shows, the simultaneous triad of low-amplitude mixed EEG, suppressed chin EMG, and bursting eye movements, is given a specific clinical name. It is called REM latency. REM latency is not simply a measurement, it is a window. In a healthy adult on an ordinary night, it falls in a fairly predictable range that's 70 to 120 minutes, reflecting the normal progression through one complete NREM cycle before the brain is ready to enter REM for the first time. The measurement says that the brain, in health, does not rush to REM, it earns it. It completes its first long descent into slow wave sleep, rises back through N2, and only then, having done that foundational work, opens the door to the stranger state that follows. When REM latency falls far outside this range, when the first REM episode appears 20 minutes or less after sleep onset, the measurement becomes clinically significant in a way that its discovery in the mid-20th century transformed the practice of sleep medicine. Severely shortened REM latency, of the kind where a person passes almost directly from wakefulness into REM with little or no NREM intervening, is among the diagnostic criteria for narcolepsy, for neurological disorder in which the boundaries between sleep states become unstable and the brain can enter REM without the usual preparation. In narcolepsy, the transition to REM is not preceded by the normal NREM descent. It arrives abruptly, sometimes in the middle of an ordinary waking moment, and the features of REM atonia, the paralysis of the skeletal muscles, can appear in a person who is still conscious, still aware, still trying to function in the waking world. Shortened REM latency is also found consistently in major depressive disorder. People in episodes of clinical depression reach REM faster than they should, in some cases within 30 minutes of sleep onset. They spend more time in REM overall, and that early REM is denser with the rapid eye movements that mark the most active portions of the state. The relationship between depression and REM architecture has been studied for decades, and the finding has proved durable across many laboratories and many populations. There are good ideas about why it occurs, involving the neurochemistry of the monoaminergic systems that regulate both mood and the timing of REM, but the full picture is not yet settled. What is settled is that the measurement of REM latency is not merely an academic curiosity. It is a number that carries clinical weight, a window into the condition of the system that produces it. The REM latency of a healthy adult on a healthy night is in this way a kind of signature of order, 70 to 120 minutes, one full NREM cycle, a complete descent and return before the stranger state is entered. The brain, given time and quiet and darkness, moves through its own architecture in a sequence that has been conserved across millions of years of mammalian evolution, and it does not skip steps. That first REM episode, when it finally appears, is the narrowest of all. It may last only 5 or 10 minutes, a brief opening at the top of the first cycle's arc, just long enough to register on the record before the NREM descent begins again. On the polysomnographic trace, it appears as a narrow band, the EEG trace quickening to its mixed low-amplitude pattern, the EMG trace falling towards silence, the EOG flickering with the first rapid movements of the night. Then all three return to the NREM configuration, and the record shows another long descent, 5 minutes, sometimes less. The first REM episode of the night is so brief that it would be easy to miss its significance, easy to see it as a hesitation rather than an arrival, a brief interruption in the NREM architecture, rather than the first instance of a state that will, by the morning's last cycle, have stretched to fill nearly an hour. But it is not a hesitation, it is the beginning of a pattern. Every subsequent cycle will return to this state, each episode longer than the last, the brain committing more and more of the late night to this particular configuration of activity, this bright, still, inwardly generating state, until the final long arc of REM, just before waking, encompasses everything the night was building toward. The architecture of the sleeping night is, in this sense, not a flat repetition, it is a progression. The 4 stages return in each cycle, and the cycles return across the night. But what the night as a whole accomplishes cannot be understood from any single cycle in isolation. The early hours and the late hours are doing different things. The deep N3 of the first cycle and the long REM of the fourth are not the same kind of sleep, even though both happen in the same sleeping body, in the same bed, in the same dark room. The night has its own arc, from the deepest biological repair of the early hours to the most active and complex neural processing of the late ones, and that arc is not visible from inside it. Only the record shows it whole. For most people, on most nights, the full architecture runs its course without any awareness that it is running. 4 or 5 complete cycles, each one 90 to 100 minutes long, each one including all 4 stages in sequence, each REM episode a little longer than the one before it. The body lies still, the room is dark, and the brain, in the hours between closing the eyes and opening them again, moves through more states of consciousness than most people have words for, from the soft dissolution of N1 to the deepest electrical silence of N3, from the brief first REM to the long final one, following an order that was not chosen and does not need to be maintained because it maintains itself night after night in every sleeping body, with the quiet reliability of something very old and very well designed. The measurements that describe this architecture, the percentages, the cycle durations, the timing of each stage's dominance across the night, were not always known. The technology to make them was not available until the mid-20th century, and the picture of what a normal night of human sleep looks like in quantitative detail was assembled gradually across decades of work in sleep laboratories on several continents by researchers who connected sleeping volunteers to machines and watched the records accumulate through the night. What they saw, slowly, was a system of remarkable regularity, not rigid. The individual variation is real, and no 2 nights are identical, even for the same person, but reliably ordered. The stages follow each other in the same sequence. The slow wave dominance in the early cycles and the REM dominance in the late ones repeat with enough consistency to be described in textbooks and taught in medical schools as normal. The range of healthy REM latency, 70 to 120 minutes, holds across populations and cultures and ages, at least in adults. That orderliness, the orderliness of the sleeping night, is one of the most consistently overlooked facts about the biology of sleep. The body's waking life is full of variation, the times of meals, the duration of activity, the level of stress, the shifting chemistry of moods, but the sleeping architecture, once properly entered, has a shape. It is not random. The brain does not wander aimlessly through the night, landing in different states by chance. It follows a path, the same path, or one very close to it, every night, a path laid down in the nervous system long before any particular human was born, running on machinery so old that its basic form is shared with the shrew and the seal and the great whale sleeping at the surface of the ocean, its breath slow and its long body still. The room is still dark. The body on the bed has not moved. The breathing is slow and even, the hands still open. The clock on the wall, if anyone were watching it, would show that roughly an hour and a half has passed since the eyes closed, since the first soft loosening of N1, the first gentle release of the room's sounds from their meanings, the first crossing of that permeable border between the lit world and the biological night. An hour and a half, and the first complete cycle is almost done. The brain has descended through N1 and N2 into the deep electrical tide of N3, has risen back, and is now approaching the end of its first full revolution. Somewhere in the brainstem, in a cluster of neurons, small enough to fit in the space of a thumbnail, the conditions for REM are assembling. The neurochemical balance is shifting. The monoaminergic systems are pulling back. The cholinergic systems are beginning to stir. The machinery that generates the strange bright state of REM is preparing, without instruction, to do what it does every night. And in approximately the next 10 minutes, measured precisely in a clinical record, as the interval between the polysomnographic signature of sleep onset and the first appearance of the REM triad, the first REM episode of this particular night will arrive. It will last only a few minutes. Then NREM will take the body back down again, and the cycle will begin its second revolution. But the interval itself, that 70 to 120 minutes, that REM latency, that quiet elapsed time between the first loosening of the border between wakefulness and sleep and the first appearance of the night's strangest state, contains, in its careful, reliable length, something of the whole shape of the sleeping night. It is the time it takes the brain to do what it needs to do before it is ready for REM. It is the duration of one complete arc from N1 to N3 and back, the foundation that every subsequent REM episode rests on, the preparation that the night's architecture requires before the stranger work can begin. That interval is the same tonight as it was last night, and the night before, in this body, and in nearly every healthy sleeping body on the planet. It has been the same for as long as there have been brains that sleep in cycles. It runs in the dark without the sleeper's knowledge, as steady and self-regulating as a tide, the hidden orderliness beneath every ordinary night of sleep. At the base of the skull, where the spine meets the brain and the bones of the cranium curve inward and down, there is a structure so small that it fits within the hollow of a closed hand. It is ancient. In the long history of the nervous system, it was here before the cortex arrived above it, before the great folded surface of the brain grew outward and complicated everything. The brainstem sits at the crossing point between thought and body, between the mind that asks questions and the organs that do not wait to be asked. And deep within it, in a region called the pons, something stirs before every REM episode begins. The pons is roughly 1.5 inches long. It sits in the dark behind the face, behind the jaw and the back of the throat, enclosed in bone and insulated by fluid. No light reaches it. No sound that the ears would recognize moves through it. What moves through it are electrical signals and the slow chemical tides that shape which signals are permitted and which are silenced. It is in this place, unhurried and invisible, that the command to enter REM sleep is assembled and issued, not from the cortex above, not from the parts of the brain associated with thought or language or intention, but from here, in the base of everything. Before a sleeper enters REM, in the minutes just prior to the transition, certain neurons in the pontine brainstem begin to increase their firing rate. They are small cells. There are not many of them. Their names reflect the geography of the pons rather than any easily pronounceable label. They are concentrated in the sublater dorsal nucleus and in two closely associated structures called the pedunculopontine tegmental nucleus and the later dorsal tegmental nucleus. None of these clusters is large enough to see without a microscope. Together they form the origin point of the REM state, and when they are active, REM follows. The firing of these cells is not random. It is not an alarm going off. It is more like a slow building of coordinated activity in a system that has been waiting, a gradual increase in the rate of electrical discharge that climbs in the minutes before each REM episode, reaching its highest level once the episode is fully underway, and then falling again as the episode ends and the brain descends back into the quieter terrain of non-REM sleep. Each REM period across the night traces this arc. The cells climb, they sustain, they fall, and because they do, the sleeper's brain enters one of the most active states in its entire biological range. What these neurons release is acetylcholine. It is a small molecule, one of the oldest signaling chemicals in the nervous system, and during REM sleep, the cholinergic cells of the pedunculopontine tegmental and later dorsal tegmental nuclei release it in concentrations that fall almost to zero during slow-wave sleep, but rise substantially as REM approaches. Acetylcholine carries the instruction outward to the thalamus, to the brainstem's own reticular formation, and eventually upward toward the cortex. Its effect, broadly, is desynchronization, where the slow-wave sleep that precedes it is marked by large synchronized, rolling electrical waves sweeping the cortex in regular rhythms. The cortex under acetylcholine's influence becomes quick and varied, and low amplitude, more like waking than like sleep, at least in the frequency profile of its electrical activity. This is one of the defining paradoxes of REM sleep. The brain enters a state that electrically looks like wakefulness, while the body remains deeply asleep. The EEG of REM, the pattern of electrical activity recorded from the scalp, is fast and mixed, similar in many ways to what would be recorded from a fully alert person. The muscles, at the same time, are held in profound silence. These two things happen together. One is not possible without the other, not in the ordinary REM state, and the chemical signal that sets the whole process in motion is acetylcholine, flowing from a small cluster of cells in the base of the brain. The role of acetylcholine in generating this state was clarified in part through animal experiments that remain instructive even decades later. When researchers delivered a chemical called carbachol, a compound that mimics acetylcholine's action at muscarinic receptors, directly into the pontine reticular formation of a sleeping animal, a REM-like state followed. The animal's EEG shifted toward the desynchronized pattern. Muscle tone fell, eye movements began. This happened within minutes of the injection in an otherwise sleeping animal that would not have entered REM on its own for some time. What the experiment demonstrated was a kind of chemical legibility in the system. There is a specific place in the brainstem and a specific chemical signal, and when that signal arrives at that place in sufficient concentration, a complete REM-like state assembles itself from the available biology. The signal does not act alone. For REM to begin, something else must change. Something must step back and allow the cholinergic neurons to act. What steps back is a set of neurons releasing entirely different chemicals, serotonin and norepinephrine. These cells belong to what are called the monoaminergic systems. The cells releasing serotonin are concentrated in a long, thin structure along the brainstem's midline called the dorsal raphe nucleus. The cells releasing norepinephrine are clustered in a small region higher in the brainstem called the locus coeruleus, a name that means blue place, after the faint pigmentation visible in fresh tissue. Both of these populations are active during wakefulness. During non-REM sleep, they slow. During REM, they fall to the lowest firing rates measurable in the brain, rates so close to zero that the transition from non-REM to REM registers in both systems simultaneously as something close to silence. Serotonin's concentration in the brainstem drops. Norepinephrine's concentration drops. The chemical environment of the pons during REM is in this sense a clearing, a space from which the two most consistently present modulatory signals of the waking brain have been withdrawn, leaving the acetylcholine system room to work uncontested. These are not incidental details. The withdrawal of serotonin and norepinephrine during REM is not merely a byproduct of the state, it is a necessary condition for it. When monoaminergic tone remains elevated, whether through the pharmacology of certain antidepressants that prevent serotonin from being reabsorbed, or through the biology of conditions that dysregulate these systems, REM sleep is suppressed. The relationship runs in the other direction as well. The monoaminergic cells during REM are held in silence, not passively, but through active inhibition from the same populations whose activity defines the REM state. The two systems are not merely alternating, they are mutually suppressing each other, each holding the other down when it is dominant, each releasing when the other rises. This mutual suppression is the heart of what the neuroscientist Clifford Saper and his colleagues described in 2001 as the flip-flop switch. The name comes from electronics, where a flip-flop circuit is a bistable device, a system with two stable states and no comfortable middle. It does not sit between states. It is in one state or it is in the other, and the transition between them, when it comes, is not a gradual drift, but a sudden decisive commitment. Either the circuit is fully in the first position, or it is fully in the second, and the crossing from one to the other happens sharply at a threshold. The brainstem circuit that governs the transition between non-REM and REM sleep works in the same way. On one side of the switch, the monoaminergic REM-off populations, serotonin from the raphe, norepinephrine from the locus coeruleus, fire steadily, and their combined output suppresses the REM-on cholinergic neurons in the neighboring nuclei. The REM-on cells are held down. REM does not begin. On the other side of the switch, the cholinergic and related REM-on populations are releasing enough signal to suppress the monoaminergic cells in return, and the brain is in REM. The transition from one state to the other is a tipping, a moment when mutual inhibition shifts, one side releasing its grip before the other has fully established its dominance. Because the circuit is bistable, intermediate states are unstable. The system does not prefer the middle. What this predicts, and what polysomnographic recordings confirm, is that the boundary between non-REM and REM appears as a sharp line rather than a gradual gradient. The EEG changes from slow wave to desynchronized within seconds, not minutes. Muscle tone drops quickly. Eye movements begin. The sleeper does not hover between states. The brain commits. Below the flip-flop switch and its cholinergic trigger, a broader network of neurons coordinates the individual features of the REM state. This network is called the pontine reticular formation, a diffuse meshwork of cells distributed through the brainstem's core, whose Latin name simply means net-like formation in the bridge. It is not a tightly bounded nucleus, but a distributed system. its cells projecting outward in many directions at once. From the pontine reticular formation, signals travel upward to the thalamus and cortex to produce the desynchronized EAG. Signals travel downward to the spinal cord and brainstem motor nuclei to produce the muscular stillness that accompanies the state. Other signals generate the bursts of rapid eye movement that give REM sleep its name, driving the lateral and vertical excursions of the eyes, visible to any careful observer watching a sleeping person's closed eyelids. Crucially, these features do not require each other. Optogenetic experiments in which specific populations of neurons are activated or silenced using beams of light delivered through implanted fibers have shown that cortical desynchronization, rapid eye movements, and the suppression of motor output during REM can be partially separated from one another when different components of the pontine reticular formation are targeted independently. The features of the REM state arrive together under normal conditions, not because they are generated by a single undifferentiated mechanism, but because the same switch activates multiple parallel pathways that ordinarily run in concert. The flip-flop switch opens the gate. The reticular formation distributes the consequences outward along distinct routes, each producing a different piece of what we observe as the complete REM state. The region of the pontine reticular formation most directly involved in this distribution is a cluster of primarily glutamatergic neurons called the nucleus reticularis pontus aurelis, a name that translates roughly as the oral part of the pontine reticular nucleus, referring to its position near the mouth-facing portion of the pons. These are not acetylcholine-releasing cells. Where the cholinergic neurons of the adjacent tegmental nuclei provide the initial trigger, the signal whose arrival sets the transition in motion, the glutamatergic cells of this reticular nucleus provide the amplification and downstream distribution. They receive the cholinergic signal and relay it outward to the structures responsible for each feature of the REM state, their activity sustaining the whole configuration throughout the episode's duration. This layering of chemical signals, acetylcholine, initiating, glutamate distributing, and sustaining, is typical of how the brainstem organizes complex states. No single molecule carries the full instruction. No single nucleus acts alone. Instead, one chemical signal opens a door, and through it flows a broader cascade of activity that reaches the thalamus and the cortex and the motor pathways simultaneously, each pathway responding with the particular feature it is built to produce. The result is a state assembled from many components, each with its own cellular origin, each contributing a different thread to the complete fabric of REM sleep. The thalamus, receiving signals from the pontine reticular formation, plays a relay role in the EEG desynchronization of REM. During non-REM sleep, the thalamus is in a particular operational mode. Its relay neurons fire in rhythmic bursts, rather than in the sustained, single-spike manner they use during wakefulness, and this bursting produces the characteristic slow waves and sleep spindles of non-REM sleep. During REM, the thalamus is shifted back out of this bursting mode, its relay cells returning to the tonic single-spike firing pattern that resembles wakefulness and that allows the rapid, desynchronized signals of REM to pass through to the cortex. The brainstem, in sending its activation signals upward, effectively releases the thalamus from the mode that had been holding slow-wave sleep in place, and the cortex above it follows. What travels from the thalamus to the cortex during REM is not random noise. The signals include regular bursts of high-amplitude electrical activity called pontogeniculo-occipital waves. Sharp potentials that originate in the brainstem pons travel to the thalamus's lateral geniculate nucleus, which handles visual information in waking and then continue on to the occipital cortex at the back of the brain. These waves were first described in cats and later identified in other mammals, and their timing correlates closely with individual rapid eye movements, each burst tending to arrive just before or at the moment when the eyes make their lateral excursion under the closed lids. In humans, they are harder to record directly, given the depth of the structures involved, but the same timing relationship between eye movements and bursts of cortical activity has been observed, and the anatomical pathway from brainstem through thalamus to occipital cortex is the same one used by real visual input arriving from the eyes during waking. During REM, that pathway is being activated from within. The signal does not come from the world. No light is entering through closed eyelids in a darkened room. The input driving the visual cortex during REM arrives from the brainstem, traveling inward rather than outward, using the same anatomical infrastructure that ordinarily carries perception, but delivering activation that the visual cortex generates from its own interior. This is part of what makes the REM state unusual in the spectrum of brain states. It recruits the sensory processing architecture of waking life, the visual cortex, the thalamic relay, the eye movement circuits, and runs them on internally generated signal rather than external input. The dorsal raphe nucleus sits along the brainstem's midline like a thin stripe of tissue. In waking, its serotonin-releasing neurons fire at a modest, steady rate, not fast but consistent, a background tone that runs through virtually every waking moment. The firing rate increases slightly during active, alert behavior. It decreases during quiet rest. And then, as sleep deepens into the REM phase, these cells go almost entirely quiet, their output dropping toward the lowest level recorded in any physiological state. Serotonin levels in the forebrain and brainstem fall accordingly. The drop is not small, it is a substantial withdrawal, a near disappearance of a signal that during wakefulness is so pervasive and constant that it often goes unnoticed precisely because it is always present. The locus coeruleus behaves similarly. Its norepinephrine-releasing neurons are active during wakefulness, increasing their firing in response to novel or salient stimuli and contributing to the alerting and orienting responses that keep a waking animal responsive to its environment. In non-REM sleep, they quiet somewhat. In REM, they reach their nadir, a near total cessation of firing, the lowest point in the day's cycle of norepinephrine release. The two systems, serotonin and norepinephrine, the dorsal raphe and the locus coeruleus, withdraw together in parallel as if following the same score, and in their withdrawal, the chemical conditions of the REM state are established. A small node in the brainstem, called the ventrolateral periaqueductal gray matter, also contributes to the suppression of the monoaminergic populations during REM. It receives input from both REM-on populations and from structures higher in the brain, and it projects inhibitory signals back onto the raphe and coeruleus in ways that help lock the monoaminergic systems into their low-firing state once the REM transition has begun. The flip-flop switch, in this sense, is not simply a two-population mutual inhibition, but a small network of structures that together maintain the bistability, ensuring that once the transition to REM has been made, it does not immediately reverse, and that once the transition back to non-REM has been made, REM does not immediately re-establish itself. This stability is what gives the REM episodes of the night their coherent duration. They do not flicker on and off in rapid succession. Each episode, once established, persists for a period measured in minutes, short in the first cycles of the night, extending considerably in later cycles, with the brain spending more and more of each cycle in the REM state as morning approaches. The circuit that switches REM on also maintains it, holding the state against the constant pressure of the monoaminergic systems that will eventually reassert themselves and return the brain to non-REM or to waking. There is a way of imagining this circuit that stays close to its physical reality. In the dark of the sleeping brainstem, two populations of neurons are engaged in a slow, sustained contest. One population holds a chemical suppression over the other. The other responds with its own suppression in return. For most of the night's early cycles, the monoaminergic population wins, or more precisely, the balance of inhibition remains in its favor, and the REM-on circuits stay subdued. As the night progresses and the early morning approaches, something shifts in the balance. The accumulating influence of circadian and homeostatic processes tilts the scale, and the REM-on populations gain ground, holding their dominance for longer and longer intervals before the monoaminergic populations reassert themselves and the episode ends. This is why REM is not evenly distributed across the night. The first REM episode, appearing after the initial 90 to 110 minutes of non-REM sleep, may last only a few minutes. The last REM episode, in the fourth or fifth cycle of a full night, may run for 45 minutes or longer. The switch operates each time with the same mechanism, the same mutual inhibition, the same sharp transition, the same cholinergic trigger, but the context in which it operates shifts with the time of night, and the duration of each REM episode reflects that shift. The cholinergic cells of the pedunculopontine and later dorsal tegmental nuclei are not only involved in generating REM. During wakefulness, they also fire at elevated rates and contribute to the cortical arousal and thalamic gating that keep the waking brain responsive and alert. Their activity during REM is, in some sense, a kind of reinstated waking signal, not identical to the waking state because the context is different and the accompanying activity of many other systems is different, but chemically and electrically related to it in ways that explain why the EEG of REM looks so similar to the EEG of wakefulness. The cholinergic systems are not specific to sleep. They do not belong exclusively to the night. What makes their activity during REM unusual is the context in which it occurs, the simultaneous suppression of monoamines, the disconnection from sensory input, the presence of atonia, and the reconfigured relationship between brainstem signal and cortical response. It is worth pausing in this picture. The brainstem, a structure often described as primitive in the sense of being evolutionarily old, is not running a simple program. What it is doing during the initiation and maintenance of REM sleep involves a finely balanced interaction among multiple populations of neurons, multiple neurotransmitter systems, multiple feedback loops, and a timing mechanism synchronized to the broader circadian and homeostatic rhythms of the body. This does not require attention. It does not require intention. It requires only that the structures are intact and that the chemical environment of the sleeping brain follows its ordinary course. When they are intact, and when it does, the same sequence unfolds every night. The monoamines falling, the cholinergics rising, the reticular formation distributing its signals, the thalamus shifting mode, the cortex desynchronizing, and the REM state assembles itself from the bottom up, every time in the dark. The pontine brainstem has no access to what the cortex above it will do with the state it creates. It does not know what the visual cortex will construct when the PGO waves arrive. It does not know what the hippocampus and the limbic structures will do with the conditions it establishes. It sends its signals. The signals propagate upward through the established pathways, and what happens in the brain above it is the consequence of its activation, not its intention. The brainstem is not dreaming, it is not imagining, it is switching a circuit on, maintaining a chemical environment, and distributing a set of signals that the rest of the brain inherits and works with. The Carbachol experiment, seen in this light, carries a particular clarity. A single compound, placed in a single small region of the brainstem, produces a complete REM-like state in minutes. The reason this is possible is not that Carbachol is a complex signal carrying many instructions. The reason is that the receiving tissue, the pontine reticular formation and its connections, is already organized to respond in a specific and elaborate way to the chemical environment that acetylcholine creates. The trigger is simple, the response is not. Everything that follows from the trigger is already latent in the architecture of the circuit, waiting for the chemistry to shift. The locus cerebellosus, lying close to the fourth ventricle in the upper pons and lower midbrain, is one of the more precisely studied structures in sleep research because its norepinephrine output reaches virtually every other region of the brain. Fibers from the locus cerebellosus project to the cortex, to the hippocampus, to the amygdala, to the spinal cord, to the cerebellum, a single small nucleus with outgoing connections that span the entire central nervous system. The withdrawal of norepinephrine during REM is therefore not a local event. It is a system-wide change in the chemical environment of the brain, a global modulation affecting every region that the locus coeruleus ordinarily reaches. The consequences of this withdrawal are not limited to any single circuit. Similarly, the dorsal raphe phase withdrawal of serotonin during REM is a global event in the forebrain. Serotonin modulates mood, arousal, social behavior, sensory processing, and a long list of other functions during wakefulness. Its near disappearance during REM removes a tonically present influence from nearly every cortical and subcortical region it ordinarily touches. What the brain experiences during REM, the specific quality of its activity, is not only a function of what the cholinergic systems are adding, it is also a function of what the monoaminergic systems are no longer providing. The flip-flop switch that Saper and colleagues described is, in this sense, not merely a switch between two states. It is a switch between two chemical worlds. In one world, serotonin and norepinephrine are present in their waking concentrations, and the brain operates within the modulation they provide. In the other world, those concentrations have fallen to near zero. Acetylcholine is dominant, and the brain operates in a fundamentally different chemical context, one that shapes how every region processes the signals it receives. The flip-flop switch is not just a gate on one piece of behavior. It is the opening and closing of two different chemical environments, each with its own downstream consequences for the entire brain. A useful way to hold this is to think of the transition into REM not as a single event, but as a clearing followed by a flooding. First, the monoaminergic systems step back, serotonin from the raphe, norepinephrine from the coeruleus, and in their withdrawal, the brainstem's chemical landscape is cleared. The acetylcholine that the cholinergic REM-on neurons have been releasing, suppressed during non-REM by the presence of the monoamines, is now free to act on its target receptors without interference. The clearing enables the flooding. The acetylcholine floods the pontine reticular formation and the thalamic relay nuclei. The thalamus shifts from burst firing to tonic firing. The cortex desynchronizes. The brainstem distributes its signals downward to the motor system and upward to the visual cortex. The eye movements begin. The REM state is established, and throughout all of it, the person sleeping in the dark is completely still. The brainstem does not announce what it is doing. It leaves no visible sign on the sleeping face, no change in the breath deep enough to notice from across a room, no sound or motion that would distinguish this moment from any other moment of deep sleep to an observer not equipped with electrodes and recording equipment. The transition from non-REM to REM, sharp, decisive, the product of a switching circuit doing exactly what switching circuits do, is entirely interior. The world outside the skull does not change when it happens. The room remains dark and still. The body remains motionless on its surface. And in the base of the brain, in the small old structures below the thinking cortex, the night's next REM episode begins to unfold, as it has every night since the brainstem was fully formed, without effort and without hesitation, committing entirely to one state, and then, when the time is right, committing entirely to the other, with the clean decisiveness of a circuit that does not linger between positions. That quality, the abruptness of the transition, the absence of any prolonged intermediate, is what the flip-flop switch model was built to explain, and it is what the polysomnographic record confirms night after night in sleep laboratories around the world. The border between non-REM and REM is a line, not a gradient. The brain on one side of that line is in a different chemical world from the brain on the other side, and the crossing between them takes only seconds. The mutual inhibition of the two populations tips. One side releases, the other rises, and the state is new. There is something in this precision that sits quietly alongside everything the brain will do with the state once it has been entered. The images that may form in the visual cortex, the emotional tone that may rise from the limbic structures, the memories that may surface and shift in the hours before waking. All of it depends on a small set of cells in the pontine brainstem doing what they have always done, measuring the chemical environment of the sleeping brain, responding to the balance of mutual inhibition, and at the right moment, releasing the circuit into its other position, with sudden complete decision. Theta waves move through the hippocampus at 4 to 8 cycles per second, not in bursts or surges, but in a steady rolling oscillation, a slow electrical tide moving back and forth through a curved structure no larger than a finger, buried deep inside the temporal lobe. This rhythm is the brain's first signature of full entry into REM sleep, the signal that something qualitatively different from all prior stages of the night has begun. It is quiet, measured, and precise, like a clock made of living tissue, running in the dark. The hippocampus sits near the center of each temporal lobe, one on the left side and one on the right, each curved gently inward like a small parenthesis. During waking life, these structures work continuously, taking in the events of the day, placing them into context, moving information toward longer storage. But during REM sleep, the hippocampus shifts into a different mode entirely. The theta rhythm that runs through it is slower than the fast oscillations of wakefulness, slower than the bursts that mark lighter sleep, and yet it carries within its steady pulse a kind of directed activity, a synchronized cycling that appears in the evidence gathered across decades of research to be doing real and consequential work. What that work is and what it requires belongs to later ground. For now, the rhythm itself is the thing, 4 to 8 cycles rising and falling every second across a structure the length of a small seashell, repeating all through the long hours of REM sleep. In rodents, where electrodes can be placed directly against the hippocampal surface, these theta oscillations are among the most robust signals in all of sleep neuroscience. They arrive cleanly, consistently, and almost exclusively during REM equivalent states. which is why researchers came to use hippocampal theta as a defining marker of REM sleep in animal work long before the full constellation of its mechanisms was understood. In sleeping humans, broad theta activity spreads across the frontal and temporal regions of the scalp in patterns recorded by electroencephalography, less confined to a single structure, distributed more widely across the cortical surface, but carrying the same fundamental frequency range and the same implication that the brain has entered its most particular nighttime state. The precise measurements vary between individuals and across laboratories, but the basic signal holds. Wherever REM sleep is found, theta is found within it, cycling at its own unhurried pace. What runs alongside the theta rhythm is something far more abrupt. Deep in the brainstem, in the region of the pons, bursts of sharp electrical activity rise with brief and powerful intensity. These potentials, high in amplitude, sudden in onset, originate in the pontine brainstem and travel upward through a specific anatomical pathway from the pons into the lateral geniculate nucleus of the thalamus, and from there into the visual cortex at the back of the skull. The path they trace is the same path that light-driven signals follow during waking vision. They travel the same road. They arrive at the same destination. And when they reach the occipital cortex, they activate it, not metaphorically, not weakly, but in ways measurable by every instrument available, driving the visual cortex into a state of real neural response in the complete absence of any visual input from the world outside the skull. Michel Jouvet first described these potentials in cats in the 1960s, and the name that attached to them reflects their path: pontogeniculo occipital waves, named for the 3 stations they pass through on their way from brainstem to cortex. In the sleep laboratory recordings of that era, they appeared as sharp spikes in the electrophysiological trace, sudden, coordinated, and tightly linked in time to the onset of rapid eye movements. They arrive just before the eyes move. They drive the movement, or something closely coupled to it, and they continue to propagate upward into the visual cortex with each new wave, each one a signal, traveling the visual pathway from the inside out. No light enters the eye during REM sleep. The eyelids are closed. The retina receives nothing from the room, and yet the occipital cortex, which processes vision, receives a steady succession of sharp electrical inputs all through the REM period, as though the brainstem were generating its own substitute for the world. These waves are one of the most consistently observed phenomena in sleep neuroscience, and their anatomy tells a clear story. The pontine brainstem sends signals upward through the same thalamic relay that carries visual information during waking. The thalamus forwards those signals to the visual cortex in the same manner it forwards light-derived signals in the day. Whatever circuits generate visual experience during waking hours are being addressed during REM sleep by a different sender, but the message is delivered along the familiar road in a language the cortex already knows. Whether this is the proximate cause of visual imagery during REM or one of several contributing factors remains a genuinely open question. What is certain is that the cortex receives the input and responds. The visual cortex during REM sleep is not simply receiving these brainstem signals in an undifferentiated way. Within the occipital lobe, there are regions that specialize, areas tuned to the processing of faces, areas responsive to motion, areas that handle color or spatial arrangement, or the angular features of objects. Neuroimaging work has shown that the activation patterns inside the visual cortex during REM are not uniform. When certain regions are more active, the area in the temporal lobe that responds to faces during waking, for instance, Dreamers who can be awakened at those moments report dreaming of faces more frequently than those awakened when that region is quieter. The same pattern appears for motion. The cortical area that processes moving stimuli during waking shows elevated activity during REM, and its level of activity correlates with how much movement appears in reported dream content. The visual cortex is not simply on or off during REM sleep. It is doing something more selective than that, and what it selects reflects, at least partially, what the dreaming mind generates in the space behind the closed eyes. None of this visual activation requires the prefrontal cortex to direct or oversee it, and the prefrontal cortex during REM sleep is doing something quite different from what it does during the day. Neuroimaging work using positron emission tomography, which measures regional cerebral blood flow as an index of neural activity, has consistently shown that the dorsolateral prefrontal cortex, the region most associated with logical reasoning, working memory, deliberate planning, bed and executive oversight, is relatively deactivated during REM sleep. Not completely silent, but measurably below its waking baseline in a way that no other broad state of the brain, waking or sleeping, quite replicates. This relative quiet is called hypofrontality. It is not incidental. It is one of the defining features of the REM brain's configuration, and its consequences ripple through everything that the dreaming mind experiences and accepts. Alan Braun and colleagues, working in the late 1990s with PT imaging, documented this deactivation clearly and proposed that it explains some of the most familiar qualities of the dreaming state: the acceptance of impossible events without question, the instability of narrative logic, the feeling of certainty that dissolves the moment waking consciousness is restored. During REM, the region of the brain most responsible for evaluating consistency, for noting that something does not follow, for maintaining a stable model of reality across time, that region is running below its ordinary capacity. What passes through the dreaming mind without scrutiny does so partly because the scrutiny mechanism is not fully available to apply. The theta rhythm continues in the hippocampus. The visual cortex lights up from brainstem signals. The prefrontal cortex rests in relative quiet, and the result is a brain that is simultaneously among the most active of the night and missing one of the components most essential to the directed, evaluated thinking that waking life requires. Alongside the prefrontal deactivation, there are regions of the REM brain that become markedly more active than their waking baseline would suggest. Pierre Mackay and colleagues at the University of Liège mapped regional cerebral blood flow across the whole brain during REM sleep using a radioactively labeled water tracer, oxygen-15, bonded to water, that distributes itself in proportion to blood flow and allows each brain region's activity to be measured in a sequence of timed scans across the sleeping subject. What emerged from that mapping was a picture that looked like neither waking nor any stage of NREM sleep. It was qualitatively its own thing, a configuration with its own logic, its own warm regions and cool regions, its own topography, as distinct as a landscape viewed from above. The regions that showed increased blood flow during REM in Mackay's work were clustered in the limbic and paralimbic system, in the brainstem, in the thalamus, and in the visual association areas. The pontine tegmentum, the brainstem structure from which REM is orchestrated, from which the eye movement signals and the PGO waves originate, was among the most consistently activated regions in the entire brain during REM sleep. The thalamus, which relays sensory information during waking, in which the pontine brainstem accesses during REM to propagate its signals upward, was warm with activity. The amygdala, embedded in the medial temporal lobe just ahead of the hippocampus, was running at levels that matched or approached those seen during emotionally charged waking experience. The anterior cingulate cortex, a region bridging the limbic and frontal systems and involved in the processing of emotional salience and conflict monitoring during waking, was active at levels above resting baseline. Visual association areas, the cortical regions that build the more complex features of visual scenes, from the raw input processed by primary visual cortex, showed elevated flow. Set against this warm activity in the limbic and visual regions, the prefrontal and parietal cortices appeared comparatively cool. The map was not simply one of global activation or global suppression. It was a selective redistribution, as though the brain, freed from the demands of navigating the external world, had rerouted its energy toward an entirely different set of priorities, running the emotional and the visual and the brainstem-driven machinery at high capacity while setting the logical, evaluative, and spatially ordered machinery into a lower gear. The whole picture assembled itself, as Mackay's group described it, as a configuration that belonged only to REM sleep, not a reduced version of waking, not a simpler version of the brain, something else entirely. The thalamus in this picture is not a passive relay. During REM sleep, it does not simply wait for sensory signals from the outside world to pass through it. It participates actively in the propagation of internally generated signals. The PGO waves from the pontine brainstem travel through the thalamic lateral geniculate nucleus on their way to the occipital cortex, and the thalamus also plays a role in modulating the theta oscillations that run through the hippocampus and broader cortical regions during REM. The thalamus during REM is running, and running with purpose, in the sense that its activity produces real and measurable effects on the cortical regions it connects to. The lateral geniculate nucleus acts as the relay for the brainstem's visual signals, forwarding them upward in the same mode it uses for real light during waking. The thalamus as a whole becomes part of the machinery that generates the REM brain's particular internal environment, not a gate standing between the outside world and the cortex, but a participant in the construction of the brain's own night world from within. When all of these features are held together, the theta rhythm in the hippocampus, the PGO waves traveling from brainstem through thalamus to visual cortex, the elevated activity in the limbic system, and the anterior cingulate and the visual association areas, the relative quiet of the dorsolateral prefrontal cortex, what emerges is a picture of a brain that has not simply powered down for the night. It has reconfigured the components that were most active during the day. The frontal systems that plan and evaluate and maintain the strict ordering of logic have stepped back. The components that respond to internal signals, visual, emotional, self-referential, have moved to the foreground. The brain has not gone dark. It has turned inward and lit a different set of rooms. The default mode network is among the most important of those rooms. During waking, it is most active when the mind is not engaged with an external task, when attention turns away from the environment and toward memory, toward the self, toward the kind of internally generated mental activity that involves recollecting the past or imagining the future. It is a network distributed across several brain regions: the medial prefrontal cortex at the front of the brain, the posterior cingulate cortex toward the back, and the angular gyrus at the junction of the temporal and parietal lobes. These regions are functionally connected. When one is active, the others tend to be active in coordination with it, and together they form what researchers have described as the brain's resting state default, the activity that persists when all else is stripped away. During REM sleep, this network shows a pattern of elevated connectivity that resembles its configuration during wakeful autobiographical memory retrieval. The medial prefrontal cortex, the posterior cingulate, and the angular gyrus come online together in a coordinated pattern, and the degree of that coordinated activity correlates, in studies where sleepers are awakened for dream reports, with the coherence and emotional resonance of what they recall. The default mode network during REM sleep is not simply idling. It is doing something with the organizational character of self-referential, inward-facing cognition, the kind of processing that, during waking, the mind uses when it is assembling a story about itself, when it is reaching back through memory and forward into anticipation, when it is constructing the sense of a continuous self moving through time. This is a striking finding, and one that holds up across multiple research groups and several different imaging methodologies. The default mode network's pattern during REM resembles its pattern during one of the most specifically self-referential kinds of waking mental activity, something that the brain normally reserves for the inward moments of a conscious, aware mind. Turning its attention to its own history and narrative appears to be running at the network level across the long hours of REM sleep each night. The precise significance of this for what REM sleep produces in the way of experience is a question that leads into territory beyond what the network patterns alone can answer. But the pattern itself is there, in the imaging records, reliable and consistent across subjects. Within the hippocampus, running alongside the theta oscillations that define REM in animal recordings, there is activity that connects the hippocampus to the default mode network's nodes. The medial prefrontal cortex and the hippocampus are anatomically linked in ways that are important during waking memory retrieval. Signals moving in both directions between these structures are involved in calling up and contextualizing episodic memories. During REM sleep, both structures show activity, and the functional relationship between them is not severed. The hippocampus, with its theta rhythm cycling steadily at 4 to 8 times per second, and the medial prefrontal cortex, lit as part of the default mode network's coordinated activity, are communicating during REM in patterns that share at least a structural resemblance to what happens when the waking mind reaches deliberately into the past. Whether that resemblance points to a shared mechanism or merely a shared frequency is not yet fully resolved. The question is a live one, and the evidence for it continues to accumulate in sleep laboratories, where the human brain is watched through the long hours of the night. The anterior cingulate cortex occupies a position in the REM brain's activation map that is worth dwelling on. During waking, the anterior cingulate is involved in the monitoring of conflict, the detection of mismatch between expectation and outcome, between intention and result. It sits at the border between the limbic system and the frontal cortex, receiving input from emotional processing regions and forwarding information toward areas responsible for behavioral adjustment. During REM sleep, it shows elevated blood flow. It is one of the warm regions in Mackay's map, active in a brain where the dorsolateral prefrontal cortex is running quiet. This combination, the anterior cingulate, engaged, the executive prefrontal regions, relatively suppressed, produces a configuration in which the brain may be tracking something like salience and emotional relevance without the full machinery of rational evaluation available to act on what it finds. The monitoring capacity, the corrective logic runs at reduced capacity. The result is a brain that may respond to the internal signals of REM with something like attention, but without the anchor of deliberate critical assessment. The visual association areas that Mackett's group found elevated during REM sleep extend beyond the primary visual cortex. They include the regions of the occipital and temporal cortex that process the higher-level features of visual scenes, the organization of objects in space, the recognition of complex forms, the integration of visual features into coherent percepts. These areas receive their input from the primary visual cortex, which in turn is being activated by the PGO waves propagating up from the brainstem. So the chain runs from the deep brainstem through the thalamus into the primary visual cortex, and then forward into the association areas that build richer visual representations from the raw signals. Each link in that chain is active during REM sleep. Each level of the visual hierarchy receives input from the level below it and processes it and passes something upward and forward. The whole visual processing hierarchy, from its thalamic entry point through its most sophisticated cortical elaborations, runs during REM on signals that the brainstem generates itself in the dark, in the absence of light or objects or any external visual world. What is particularly clear in the overall picture that emerges from both blood flow mapping and EEG-based source analysis is that REM sleep's brain activity is not a diminished version of wakefulness. It is a different state with a different geography. The regions that dominate during waking, the prefrontal cortex managing goals and logic, the parietal cortex maintaining spatial attention, and the sense of a body located in space, are quieter. The regions that operate at high intensity during REM, the limbic system, the visual cortex, the brainstem tegmentum, the anterior cingulate, the default mode networks nodes, are running at levels that during waking would correspond to active emotional processing, active visual experience, or active self-referential thought. The brain has not rested from its business. It has changed its business entirely, and the change is orderly and specific, governed by the same cellular mechanisms that produce every other feature of the REM state, arising and dissolving on a schedule that repeats across every cycle of every night. The EEG recorded from the scalp during REM sleep has a surface appearance that is, in some ways, deceptively similar to waking. The high-amplitude slow waves of deep NREM sleep are gone. In their place is a low-amplitude signal of mixed frequencies, some theta, some alpha, some faster beta range activity woven through that at first glance resembles the desynchronized, relatively low-amplitude signal of relaxed wakefulness. Early researchers, encountering this pattern alongside the rapid eye movements and the story of atonal paralysis, called it paradoxical sleep, a term still used in European and especially French language neuroscience, because the brain looked electrically awake while the body was more deeply still than in any other sleep stage. The paradox is not a contradiction once the architecture beneath the surface is understood. The EEG's mixed frequency appearance during REM reflects genuine cortical desynchronization, billions of neurons not swinging together in the slow unified rhythms of NREM, but running in smaller, faster, more locally organized patterns. The net result on the scalp is low amplitude and apparent disorder. Within local circuits, there is real coordination. The theta rhythm in the hippocampus is one example. A genuine local oscillation, robust and regular, occurring within the broader landscape that looks from the scalp surface like something close to noise. The distinction between the scalp EEG and the activity within specific brain regions matters because it shapes how the REM brain is understood. The surface record during REM shows low-amplitude desynchronized activity. The interior shows structured, coordinated oscillations, theta in the hippocampus, gamma band activity in certain cortical regions, the sharp synchronized propagation of PGO waves across the visual pathway, the coordinated activity of the default mode network's distributed nodes. The surface looks like waking. The interior is something else, organized differently, running different computations, lit in a different pattern than any waking state produces. Broad theta activity spreading across the frontal and temporal lobes of the human scalp during REM sleep shows correlations with specific cognitive and emotional outcomes. Subjects who show more robust theta during their AM periods, in the sense of greater power at theta frequencies across the frontal scalp, tend to show better performance on certain emotional memory tasks the following day, better recognition of emotional material presented before sleep. The connection is not perfectly understood at the mechanistic level, and the path from scalp theta to the specific neural events that produce this advantage is not yet fully mapped, but the correlation is consistent across multiple research groups and carries with it the implication that theta during REM is not simply an oscillation accompanying the state, but one that participates in what REM sleep actually accomplishes. However, the final account of those accomplishments will read when it is more completely understood. At the very back of the skull, where the occipital lobe lies close to the surface, the visual cortex receives the brainstem's PGO wave propagation and responds with activity that climbs and falls across the REM period in coordination with eye movements. Each burst of rapid eye movement, visible in the electrooculographic record as jagged deflections, the eyes tracking invisible objects behind the closed lids, is preceded by a PGO wave arriving at the occipital cortex from the brainstem. The timing is consistent. The brainstem fires. The thalamus forwards the signal. The visual cortex activates. The eyes move. The sequence repeats in varying intensity through the entire AM episode. When the eyes are still, in the quieter phases between movement bursts, PGO waves are still present but less frequent. The visual cortex runs at a slightly lower level. The state continues, but the phasic activity within it has settled temporarily. Then another wave arrives, and the eyes move again, and the occipital cortex climbs in response. This phasic quality, the alternation within AM between moments of more intense activation and quieter intervals, is another layer of internal structure within the state. AM sleep is not simply a sustained plateau of activity across all regions simultaneously. It contains within itself a finer rhythm, tonic REM, which is the ongoing state with its baseline of theta and desynchronized cortical activity, and phasic REM, which is the set of short-duration events, the rapid eye movements, the PGO waves, the accompanying surges in limbic and visual cortex activity that punctuate the tonic background at irregular intervals. Both tonic and phasic REM are part of the same state. They share the same polysomnographic signature, the same brainstem neurochemistry, the same global absence of the monoaminergic tone that characterizes waking. But within that shared state, phasic events drive sharper and more intense activation in the regions receiving the brainstem's signals, while tonic intervals represent a somewhat less intense, but still distinctly REM configured neural environment. The distinction between phasic and tonic REM has practical implications for what is recorded in awakening experiments. Dreamers awakened from phasic REM tend to report richer, more visual, more emotionally intense content than those awakened from tonic intervals, consistent with the idea that phasic events are the moments when the brainstem's activation of the visual and limbic systems is at its sharpest. What assembles across all of this, the theta rhythm cycling in the hippocampus, the PGO waves propagating from brainstem to thalamus to occipital cortex, the elevated activity in the limbic system, and anterior cingulate and visual association areas, the relative quiet of the dorsolateral prefrontal cortex, the coordinated engagement of the default mode network, the phasic surges accompanying rapid eye movements, is a picture of a brain state unlike any other. The mammalian brain enters, not waking, not NREM sleep, not a reduction of waking or a lesser version of it, something fully organized and self-consistent with its own regional priorities, its own oscillatory signatures, its own internal timing, and its own qualitatively distinct profile of activity that no other state replicates. The REM brain is a brain whose external sensory gates are largely closed, whose motor pathways are held in suppression, whose prefrontal evaluative systems are running at reduced capacity, and whose internal generative systems, the brainstem producing signals, the thalamus forwarding them, the visual cortex responding, the limbic system running warm, the hippocampus cycling in its steady theta rhythm, are active at levels that the sleeping body's stillness and the room's quiet give no outward sign of. From the outside, a person in REM sleep lies still, breathes with slight irregularity, has eyes moving rapidly beneath closed lids, and is otherwise indistinguishable from a body at rest. From the inside, across the 3 pounds of tissue between those closed eyes and the back of the skull, an entirely different night is underway. The map of regional blood flow during REM sleep, brainstem and thalamus and amygdala and anterior cingulate and visual cortex, warm with elevated flow, the prefrontal and posterior parietal regions cooler against the baseline, is not a pathological pattern. It is not a sign of something going wrong. It is the brain's own doing, assembled from its own circuitry, produced by its own chemistry, repeating in every person who sleeps a full night with sufficient time in the later cycles when REM episodes stretch long. The configuration appears without instruction, sustains itself for the duration of each REM episode, dissolves as the episode ends, and the sleeper slides back toward NREM and then reassembles again in the next cycle, a little longer each time as the night progresses toward morning. The first REM episode of a full night may last only a few minutes. By the 4th or 5th cycle, the episode may extend to nearly an hour, a long, unbroken interval during which the whole configuration holds. The warm limbic activity, the visual cortex receiving its brainstem signals, the theta cycling, the prefrontal cortex quiet, the default mode network running in its particular coordinated pattern. And then it ends. The monoaminergic neurons that were suppressed during REM, the serotonergic cells of the dorsal raphe, the noradrenergic cells of the locus coeruleus, begin to recover their firing. The cholinergic drive that sustained the REM-on state withdraws, the EEG shifts, the eye movements cease. The hippocampal theta settles into a different regime. The regional blood flow map redistributes itself, warming and cooling in different places as NREM sleep returns and the slow waves build again. The REM configuration dissolves. Everything that was lit, the limbic warmth, the visual activation, the brainstem's propagated signals, dims back toward the baseline of the night. The brain makes the transition quietly, without the sleeper's knowledge, without any experience of the change. What persists after the episode ends is the same question that follows any careful examination of the REM brain's internal state. What does the configuration do? The activation is real. The oscillations are real. The regional pattern, distinctive, reproducible, consistent across subjects and across species in its broad features, is real. What all of it produces, in the biological sense of causing something to happen that would not otherwise happen, is a question that has occupied sleep neuroscience for decades and continues to generate evidence and debate and careful revision. The answer, whatever final shape it takes, will be built on top of this. The fact that during REM sleep, every night, the brain assembles a specific and precise configuration of activity that it generates entirely from within itself, sustaining it across long, quiet intervals while the body it inhabits lies still in the dark. The brainstem fires upward through the thalamus. The visual cortex lights from the inside out. The hippocampus cycles in its measured theta rhythm. The limbic system runs warm. The prefrontal cortex rests in its relative quiet. And the whole arrangement holds, steady and self-contained, in the depth of the sleeping night, assembling itself without instruction and dissolving without announcement, as it has in every mammal that has ever slept long enough to reach the far side of the night's first long descent. A body lies in the dark, fully still, not the stillness of a body that has paused or rested or chosen to remain quiet for a moment. Something deeper than choice has settled across every skeletal muscle, a biological silencing that works from the spinal cord outward, holding the arms, the legs, the long muscles of the back in a suspension more complete than anything voluntary effort could produce. The chest rises, the heart moves, but every muscle that a person might call upon in waking life, every fiber that reaches from bone to bone across a joint, is held in a state of near total electrical silence, while the brain, 3 pounds of tissue sealed inside the skull above, runs at something close to its full waking capacity. This is what the body does during REM sleep. It becomes, in almost every voluntary sense, a body that cannot move. And the mechanism that produces this is not a failure of the nervous system. It is one of its most precisely assembled achievements. The spinal cord carries the signal. Deep within its gray matter, arranged along its length in columns of cells that reach from the base of the skull to the lower back, are the motor neurons, cells whose long projections run outward through the peripheral nerves to every skeletal muscle in the body. When these cells are active, the muscles they serve contract. When they are silent, the muscles release. During REM sleep, they are held in silence by a chemical issued directly onto their membranes, a molecule called glycine, an amino acid that functions here not as a building block, but as a brake, locking the motor neurons into a state of electrical suppression so thorough that they cannot respond even to signals that would easily drive them during waking. The glycine arrives from inhibitory interneurons in the spinal cord, cells that receive their instructions from higher up in the brainstem. When those interneurons activate, they release glycine onto the motor neurons below, driving the membrane potential of those cells downward, away from the threshold at which they would fire. The cell is held below that threshold for the duration of REM. The muscle it serves receives no command. The limb lies still. Francisco Morales and colleagues demonstrated this with a clarity that has held for decades. In experiments using cats in natural sleep, they delivered strychnine directly into the spinal fluid. Strychnine is a glycine antagonist. It competes for the same receptors that glycine binds, blocking the inhibitory signal without triggering any of its own. The result was a partial release of motor suppression during REM sleep, with the animals showing movements that would ordinarily be absent. The muscle tone that REM should have abolished returned in fragments. The experiment did not disturb the brain's REM state. The EEG signatures remained intact, but the spinal brakes had been chemically removed, and the body responded. What that experiment showed was that the stillness of REM sleep is not a passive quiet, but an actively enforced one, held in place by chemistry that can be interrupted, and that when interrupted releases the body back into motion while the brain continues dreaming above. Glycine is not the only inhibitory signal involved. Gamma-aminobutyric acid released by GABAergic neurons in the brainstem and spinal cord contributes alongside it, and the two systems together produce inhibitory postsynaptic potentials of large amplitude across the motor neuron membrane. Intracellular recordings from lumbar motor neurons in sleeping cats captured these potentials directly, large voltage deflections arriving repeatedly, driving the membrane away from threshold with each wave. The cells were not merely quiet, they were being actively suppressed, wave after wave, for the full duration of each REM episode. The H-reflex confirms this in human subjects. The reflex arc is simple and old. A stimulus delivered to the skin over the back of the knee activates sensory fibers that carry the signal to the spinal cord. There, those fibers synapse directly onto motor neurons in the same cord segment, which activate and drive a brief contraction of the calf muscle. It is one of the most direct tests of spinal motor excitability available without surgery. In waking, the reflex fires reliably and cleanly. In NREM sleep, it is somewhat reduced. During REM sleep, it is profoundly attenuated or absent entirely. The stimulus arrives. The sensory pathway carries it to the spinal cord, and the motor neuron, held by the chemistry of REM against the wall of its own suppressed membrane potential, cannot rise far enough to respond. The reflex does not pass. The signal reaches the gate and finds it closed. That closed gate is the product of something small and specific, a nucleus in the brainstem no larger than a grain of rice, tucked into the pontine tegmentum, called the sublaterodorsal nucleus. It is not a structure a person could find without a microscope and a very precise map. It sits in the dorsal part of the pons, the brainstem segment that bridges the midbrain above and the medulla below, and its neurons send long projections downward through the spinal cord and outward to brainstem motor nuclei that control the muscles of the face and the throat. During REM sleep, these neurons become active. Their fibers reach the inhibitory interneurons in the spinal cord, activating them, setting off the chain that ends in glycine released onto motor neurons, and the motor neurons held below threshold, and the body lying still. The evidence for the sublaterodorsal nucleus as the origin of remitonia comes from lesion studies in animals whose precision has been difficult to achieve by any other method. When the sublaterodorsal nucleus is selectively damaged in rats and cats, its cells destroyed while the surrounding tissue is left intact, the animals continue to enter REM sleep. The brainstem mechanisms that produce the EEG signatures of REM, the rapid eye movements, the theta oscillations in the hippocampus, all continue to run, but the atonia does not arrive. The animals, deep in the electrical state of REM, stand up, they walk, they perform what appear to be organized sequences of motor behavior, grooming, locomotion, orientating movements, while their brains are generating a state that should render them physically motionless. The disconnect is precise. The REM state is present. The stillness that should accompany it is not. What those animals are displaying is a form of the same condition that appears in human clinical medicine, a condition first described in clinical literature by Carlos Schenk and Mark Mahowald, working in Minneapolis in 1986. They described a group of patients who had come to clinical attention for a reason their bed partners described in sometimes frightening terms. These were adults, most of them older, who during sleep would speak, shout, laugh, or cry out, and who would move, sometimes violently, in ways that seemed to correspond to some ongoing internal experience. They would throw punches, they would kick, they would leave their beds and fall. Some had injured themselves or their partners with movements delivered in complete sleep, movements that appeared purposeful and coordinated, as though acting out a physical sequence rather than simply thrashing. And in every case, polysomnographic recording confirmed that these episodes occurred during REM sleep. The EEG showed the low amplitude, mixed frequency pattern of REM. The electroencephalography showed the bursts of rapid eye movements. But the electroencephalography, which should have shown near complete suppression of muscle tone, showed instead normal or elevated muscle activity. The atonia had failed. Schenk and Mahawald named it REM sleep behavior disorder. The name is precise to the point of clinical flatness, and it does not convey what is actually being observed. What is being observed is the body doing what it would normally be prevented from doing, moving through the content of REM sleep, because the brainstem circuit that locks the motor system down has stopped working with the reliability it maintains in healthy sleep. The sublater dorsal nucleus, or the pathways downstream of it, or the inhibitory interneurons at the spinal level, are not enforcing the chemical suppression that should be in place, and so the body moves. It enacts, it responds to whatever the brain is generating during REM in the way the body would respond if it were awake and the scene were real. At the time of its description in 1986, the condition was understood primarily as a sleep disorder, a disruption of REM physiology with clinical consequences for the safety of the patient and anyone sleeping nearby. The deeper significance of the diagnosis emerged over the decades that followed, through a finding so consistent and so striking that it has changed the way neurologists understand the early stages of certain neurodegenerative diseases. The neurodegenerative diseases in question belong to a family defined by the abnormal accumulation of a protein called alpha-synuclein. The group includes Parkinson's disease, Lewy body dementia, and multiple system atrophy. Each destroys different populations of neurons by different mechanisms and at different rates, but in each of them, the misfolded protein accumulates inside the neurons of the brainstem and other regions in structures called Lewy bodies. And in each of them, the brainstem neurons that are lost or damaged in the early stages of the disease include exactly those neurons that maintain REM atonia, the cells of the sublater dorsal nucleus, and the circuits it drives. Longitudinal studies following patients diagnosed with REM sleep behavior disorder over many years and tracking which neurological conditions subsequently developed have produced a number that is difficult to hold lightly once encountered. More than 80% of people with idiopathic REM sleep behavior disorder, meaning the condition for which no other cause has been identified, go on to develop Parkinson's disease, Lewy body dementia, or multiple system atrophy within 10 to 15 years of the sleep disorder's diagnosis. The sleep disorder is not a consequence of the neurodegenerative disease. It is a predecessor, the same brainstem pathology that will eventually produce the motor signs of Parkinson's or the cognitive signs of Lewy body dementia. First makes itself known by dismantling the circuit that should hold the body still during REM sleep. The condition is now understood not as a sleep disorder that occasionally precedes neurodegeneration, but as a prodromal marker, an early sign of a disease that has not yet declared itself in any other way, that happens to be detectable in the sleep laboratory years, or even decades before the neurological diagnosis would otherwise be made. This places the stillness of REM sleep in a different light entirely. The paralysis that settles across the body during each REM episode is not an incidental feature of the state, a simple constraint to prevent the body from thrashing. It is a product of a specific neural circuit whose integrity reflects the health of a region of the brainstem that is among the earliest targets of synuclein pathology. When the stillness of REM sleep is lost, when the body begins to move through its own dreams, What that loss often signals is a change in the brainstem that has not yet reached the level of clinical visibility, but that will, over years, become a disease with a name. The body in REM sleep is quiet because a small cluster of cells in the pons is doing its work. When those cells begin to fail, the quiet is lost first. Long before the tremor, long before the shuffle, long before the forgetting, the nights begin to move. For most people, through most of their lives, the circuit holds. The body lies in full atonia, every voluntary muscle suppressed, the H reflex abolished, the inhibitory postsynaptic potentials arriving in waves across the spinal motor neurons. The brain above generates its most active nighttime state. The signals that would, in any other configuration, drive movement into the limbs are being generated. The commands are there, somewhere in the circuits above the spinal cord, but they cannot pass. The gate is closed. The body stays still, and yet within that stillness, something moves. The fingers twitch. A toe curls briefly and then relaxes. The muscles around the mouth shift in a small, brief contraction, not a full movement, not a purposeful gesture. A flicker, brief, isolated, irregular, occurring not because the atonia has failed, but as a punctuation within it. These are phasic muscle twitches, and they have a different origin and a different story than the tonic suppression of the major muscles. Mark Blumberg, working at the University of Iowa, has spent years examining what these twitches are and where they come from. They are generated by the brainstem, specifically by motor commands descending from above that briefly penetrate the spinal suppression, producing a small, isolated contraction that lasts a fraction of a second before the suppression reestablishes itself. They occur most often in the fingers, the toes, and the muscles of the face. They appear in the same temporal distribution as the bursts of rapid eye movements that define the phasic aspect of REM sleep, clustering in the periods of most active pontine output and subsiding in the quieter intervals between. In adult humans, these twitches are small and brief enough to pass unnoticed, present in the electromyographic record as brief spikes that interrupt the otherwise quiet signal of atonia, but rarely large enough to produce visible movement. In infants, the situation is different. A sleeping newborn, in the REM equivalent state that occupies more than half of its total sleep time, shows twitching at a rate far higher than any adult. The fingers, the toes, the eyelids, the corners of the mouth, the limbs themselves, all show brief, rapid twitches throughout REM sleep at a density that makes the infant's surface flicker with small movements. The overall muscle tone is still suppressed compared to waking, but these brief phasic events punctuate it constantly, more like a finely textured quiet than the flat silence of adult REMian tonia. Blumberg's hypothesis, built across years of detailed electrophysiological work in rats and other developing animals, is that these twitches are not random. They are generated by specific brainstem motor circuits that activate briefly during REM sleep, producing a movement in a specific muscle group, a wrist, a toe, the muscles controlling a finger, and then the sensory consequence of that movement. The feedback from the proprioceptors in the muscle and the joint travels back up the nervous system to the developing brain. The brain receives information about what it felt like to move that particular structure in that particular way. And over thousands of these events occurring through every REM episode, through every night of the long months of early infancy, the developing sensory motor cortex builds a map. This is, in essence, the hypothesis that REM sleep twitches are a mechanism of sensory motor learning, a way for the developing nervous system to measure itself. The brain sends a small controlled motor command, the body makes a small, isolated movement. The sensors in the moved structure report back, and the cortex, receiving that report, adjusts its internal model of the body's shape, which joint is where, how much force a particular command produces, what the spatial relationship between one finger and the next actually is when that finger moves. The map is drawn from the inside, one tiny movement at a time, while the body is otherwise held still and the external world is shut out. The infant has not yet learned its own body in any complete sense. The hands are new, the feet have never been walked on. The neural circuits that will eventually allow for reaching, grasping, walking, and all the intricate motor coordination that a human life requires have not yet been calibrated to the specific physical dimensions of the body they must serve. Blumberg's proposal is that REM sleep, and specifically the phasic twitches generated within it, provides a portion of the raw data from which that calibration is built. It is a hypothesis, and the full picture remains open, but the evidence is detailed and accumulating. Electrophysiological recordings in neonatal rats show that limb twitches during REM-equivalent sleep drive, reliable activity in the somatosensory cortex, the region that maps the body's surface and receives proprioceptive input, and that this activity is spatially organized, meaning that twitches in a forepaw drive responses in the forepaw representation of the cortex, not in some general signal that the system has been stimulated. The signal is specific. The map is being written with some precision. Against this backdrop, the proportionally enormous REM fraction of infant sleep begins to make a different kind of sense. Human newborns spend approximately 8 hours per day in REM-equivalent sleep. The proportion is at its highest at birth and declines steadily through childhood and adolescence toward the 20 to 25% of adult sleep. The developmental arc runs in parallel with the arc of sensorimotor learning. The years of highest REM proportion are also the years of the most rapid motor development, of the acquisition of reaching and grasping and walking, and the fine control of the hands. Whether the high REM fraction is causally connected to the pace of that development, or whether both simply track a common underlying developmental trajectory, is a question the field is still working through. What is clear is that the phasic twitches of REM sleep are not artifacts, not meaningless noise in the motor record. They are generated by specific circuits. They produce specific sensory signals, and those signals reach the developing cortex in a spatially organized way. Something is happening in those small flickers of the fingers and the face. The full account of what is still being written, the stillness of REM sleep, then, is not a single thing. It is layered. There is tonic atonia, the deep, sustained suppression of skeletal muscle tone that holds the body motionless throughout each REM episode, maintained by glycine released from spinal interneurons driven by the sublater dorsal nucleus. And within that tonic background, there are the phasic twitches, brief, organized motor events generated by brainstem commands that briefly penetrate the suppression and produce a small movement before the gate closes again. The atonia is not total in the sense of being textureless. It is a floor, firm and maintained, with the phasic events rising briefly above it and then settling back into silence. The eyes are the most visible instance of this layering. The rapid eye movements that give REM sleep its name are not a failure of atonia. They are its exception. The extraocular muscles which move the eyes are not fully suppressed during REM in the same way as the limb and trunk muscles. They participate in the phasic bursts that punctuate the state. During each cluster of rapid eye movements, the eyes trace quick, conjugate trajectories beneath the closed lids, upward, sideways, diagonal, in sequences that have been studied for decades in search of a relationship to the visual events of dreaming and that remain not fully decoded. The pontine brainstem generates the signals for these movements through pathways that run upward to the nuclei controlling the extraocular muscles. They are, in a sense, the most visible twitch of all, the phasic motor event that the state is named for, recurring in the same clusters as the other phasic phenomena of REM and produced by the same downstream drive from the active pontine generators of the state. The diaphragm keeps breathing, the heart keeps beating, smooth muscle throughout the body continues its work, unaffected by the atonia that has silenced the voluntary system. The distinction between smooth and skeletal muscle is not arbitrary. It reflects the functional logic of REM atonia, which targets the muscles of voluntary movement, while leaving the autonomic machinery of life running without interruption. The diaphragm itself is partially exempt, contracting in the slightly irregular pattern of REM breathing, while the rib cage muscles that would assist it in deep breathing are more substantially suppressed, contributing to the shallow, faster respiratory pattern that characterizes the state. What remains is a body organized around a very specific kind of suspension. The voluntary system is held, the autonomic system runs, the brain is active, and the boundary between what the nervous system is doing above the brainstem and what the brainstem is permitting the spinal cord to do is maintained by a circuit whose components can now be identified: the sublater dorsal nucleus, its projections to spinal interneurons, the glycinergic and GABAergic signals those interneurons release, and the large inhibitory postsynaptic potentials that hold the motor neurons at rest. It is a circuit that runs every night, in every adult, through each REM episode. It produces nothing visible. Its function is the absence of something that would otherwise be present, the movement that should follow the brain's motor signals, but does not, because the gate at the spinal level is closed. The achievement is invisible precisely because it works. The person lying in a REM atonia in a dark room shows nothing, no sign that the nervous system is performing one of its more precisely engineered acts, holding a fully active brain inside a fully still body for stretches of an hour or more at the end of each night. The circuit is old. All mammalian species studied show REM sleep with atonia, and the brainstem structures responsible for it are among the most phylogenetically conserved regions of the vertebrate nervous system. The pons that generates atonia in a rat is built along the same organizational principles as the pons that generates it in a person. The glycine released onto spinal motor neurons in a sleeping cat is the same molecule, binding the same receptor, producing the same inhibitory current as in the human spinal cord during REM. The mechanism is ancient and consistent. It arose once early and has been maintained across the full span of mammalian evolution. Why? The honest answer is that the question has no single settled answer. The most often cited functional account is protective. Without atonia, the motor commands generated by the active brain during REM sleep would drive the body into movement, and the resulting physical activity would be disruptive at minimum and dangerous at worst. The evidence from REM sleep behavior disorders supports this in a concrete way. What happens when atonia fails is exactly what this account predicts. The body enacts the brain's REM state in motion, sometimes violently, and the consequences are injurious. But the protective account, while reasonable, may not be the complete picture. Atonia does not merely prevent dreaming from becoming moving. It produces a specific physiological state that may carry its own functional significance, a state of motor quietude that exists alongside the active brain in a combination that does not occur at any other point in the sleep-wake cycle. Whether that combination has consequences for how the brain consolidates or reprocesses the information it generates during REM is an area the field has not fully resolved. What can be said with confidence is this: the atonia of REM sleep is real, it is chemically specific, it is actively generated and actively maintained, and when it fails, the failure is diagnostically important in ways that extend far beyond the sleep laboratory. The body held in that stillness through a full night of cycling, 4, 5, 6 times, through the rise and fall of the sleep cycle, each return to REM, bringing another episode of complete motor suppression, emerges in the morning, having been physically still for the better part of 8 hours, while the brain above it has run through states of activity that would register in any other organism as full waking. The stillness and the activity are not in tension with each other. They are the same state, seen from 2 different levels of the nervous system, and somewhere within that stillness, the small events continue. A finger twitches in the dark and is still. The toe of an infant curls briefly, releases, and the sensory signal travels upward through the cord to a cortex that is quietly learning the shape of the body it inhabits. The signal arrives, is integrated into some map whose outlines are still being drawn, and fades. The inhibitory wave arrives again at the motor neuron membrane. The potential drops, the cell falls silent, the night continues. The body is a thing that learns itself slowly, from within, in the dark. The largest muscles, the deepest suppressions, the most complete silencing, those are the architecture of the state, the floor that holds everything. But at the edges of that floor, in the small muscles of the hands and the face, in the flickering periphery of a system that is learning its own boundaries, something continues that is neither sleep nor waking, neither movement nor stillness, but a fine and patient process of calibration that has been running in every infant who has ever slept, and that leaves its record not in any outward sign, but in the slow accumulation of a nervous system that morning by morning knows itself a little more completely. The sublater dorsal nucleus sends its long projections down through the cord. The interneurons release their glycine. The motor neurons rest, and the small exceptions, the phasic twitches, the flickering fingers, the moving eyes beneath the closed lids, trace their brief arcs and subside, each one a message sent and received in the quiet interior of a sleeping body, folded inside the larger stillness that holds it all in place. Deep in the medial temporal lobe, buried beneath folds of cortex on both sides of the brain, there is a small structure shaped roughly like an almond, no larger than the tip of a thumb. During REM sleep, this structure burns, not with heat, but with metabolic activity, glucose moving through its cells, oxygen consumed, electrical signals cascading from neuron to neuron, with the same intensity seen during the most frightening or most exhilarating moments of a waking life. The amygdala does not rest when the body rests. It reaches its highest activity of the night, precisely when the eyes have been closed longest, when the muscles have gone silent, when the rest of the world has receded entirely, and in that quiet, it begins its work. Both amygdalae are active during REM sleep, the left and the right, one buried in each temporal lobe, flickering in the dark of the skull with a warmth that no waking observer could detect from outside. Positron emission tomography and functional magnetic resonance imaging have traced this heat in sleeping humans, mapping the blood flow and oxygen use that signal where the brain is spending its energy. And what they have found is that during REM the amygdala consumes resources at levels matching or exceeding the most emotionally provocative moments of full wakefulness. A structure small enough to hold in the palm of one hand, generating an internal emotional landscape as vivid as anything the waking world can offer. This is where the dreaming mind is colored. This is where the feeling of the dream is made, before any image forms, before any narrative takes shape. The emotional temperature of the night is set here, in these two small nodes of ancient brain tissue running quietly at their peak. The amygdala does not work alone. It is connected to the hippocampus alongside it, to the prefrontal cortex above, to the brainstem below, to the visual cortex at the back of the skull. And during REM sleep, all of these connections are active. All of these pathways are open. All of these structures are passing information back and forth in a configuration the brain does not use at any other time. The amygdala sits at the center of this network as something close to an emotional coordinator, not directing the dream so much as saturating it. The content that emerges from the visual cortex, the narrative fragments that surface from memory, the vague sense of who and where the dreaming self is, all of it passes through or near the amygdala's field, and the amygdala colors what passes. It tips experience toward feeling. It makes the dream matter, even before the waking mind can say why. Much of what the amygdala generates during REM is weighted toward the negative. Fear appears more often in dreaming than contentment. Threat is more common than safety. The emotional repertoire of the dreaming brain leans darker than the waking world would warrant, and the amygdala's disproportionate activation during REM sleep is part of the explanation. This is a structure whose evolutionary purpose is to detect and amplify threat, to hold the nervous system in readiness. And during sleep, its activation is not modulated by the calming inputs it receives during wakefulness from the frontal cortex. In sleep, the amygdala runs closer to its own nature. What it generates is not pathological. It is the brain using the quiet of the night to process the emotional weight of what the day has carried. From the brainstem, a wave is rising. It begins deep in the pontine tegmentum, in a cluster of cells that fire just before the eyes move beneath the lids, generating a sharp electrical potential that travels upward through the brain along a path that mirrors the one taken by real visual signals. The wave passes through the lateral geniculate nucleus, the thalamic relay station where visual information from the eyes is normally sorted and forwarded, and it continues upward through the white matter until it arrives at the occipital cortex at the back of the skull. There it lands, and the visual cortex receiving it responds as it would respond to light. These are pontogeniculo-occipital waves, first described in sleeping cats in the 1960s, and they are among the most precisely studied signals in the neuroscience of dreaming. The primary visual cortex activates in response to these brainstem waves. So does the secondary visual cortex. The regions responsible for processing faces, for interpreting motion, for distinguishing edges and colors, and the spatial relationships between objects. These too become active during REM sleep in the complete absence of any visual input from the outside world. The eyes are closed. The eyelids let through no meaningful light. The retina are not sending signals of any significance to the brain, and yet the visual cortex performs as though a world is present. Because from below, from the brainstem's pulsing waves, signals are arriving that are in the language of cortical cells, indistinguishable from the real thing. The imagery of the dream is not invented from nothing. It is evoked. The brainstem sends the signal, and the cortex fills in the world. What the visual cortex generates during REM is not random. Neuroimaging has found something more specific than general visual activation. When a dreamer is later able to report that a face appeared in the dream, the fusiform face area, a region of the temporal cortex specialized for face processing, shows elevated activity in the REM recording. When movement appears in the dream, The motion processing area, sometimes called area MT or V5, shows its own characteristic signature. The brain is not generating a uniform visual noise. It is generating something organized, something that has the structure of experience, something that activates the specific cortical machinery that would have been needed to see those things in the waking world. The dream looks like something because the looking is real. The neural architecture of perception is the architecture of dreaming, turned inward and run in the dark. Above the visual and limbic activity, a network of regions is coordinating something harder to name. The medial prefrontal cortex, the posterior cingulate cortex, the hippocampus, and the angular gyrus, a distributed set of structures that during wakefulness are most active when the mind turns inward. When a person is remembering the past or imagining the future, or thinking about who they are, these regions are also active during REM sleep, and their activation pattern during dreaming closely resembles their pattern during wakeful autobiographical reflection. This network is called the default mode network, and during REM sleep, it does not go quiet. It hums, it coordinates. It seems to do, in sleep, something close to what it does when the waking mind is left to itself, drawing on memory, weaving together fragments, constructing a version of the self moving through time and space. The hippocampus within this network is particularly active, situated beside the amygdala in the medial temporal lobe, curved and compact. The hippocampus holds a special relationship with memory, and during REM sleep, it appears to be doing something with the memories it holds, not consolidating them in the simple sense of fixing them in place, but passing them, sharing them, letting them surface into the dreaming narrative in forms that are rarely straightforward. A face from 10 years ago appears attached to a location from last week. An emotion from one memory colors an event from another. The hippocampus during REM is not a filing system quietly cataloging the day. It is more like a tide that moves things, a current that rearranges and combines, offering material to the dreaming network in configurations that waking logic would not have assembled. Clinical cases have offered some of the clearest evidence for the role of the medial prefrontal cortex in dreaming. Patients with lesions to this region, damage from stroke or from injury in the area just above the eyes toward the midline of the brain, sometimes report a loss of dreaming. Not disrupted dreaming, not frightening dreaming, but its near complete absence. They wake from sleep and find nothing there. The richness that most people experience every night, the faces, the places, the emotional sequences that fade before breakfast, simply does not occur. This is not because the rest of their REM machinery has failed. In many of these patients, the brainstem generates its waves, the eyes move, the body maintains its atonia. All the biological apparatus of REM sleep is present. What is absent is the medial prefrontal cortex's contribution to shaping that raw activity into something experienced as a dream. The narrative thread, the self that moves through the dream, the coherence of what is felt, these seem to require this region. Without it, the dreaming brain has its music, but no story. There is a state within REM sleep that differs from ordinary dreaming in one precise way. In it, the person who is dreaming becomes aware that they are dreaming. The dream continues, the imagery, the feeling, the narrative, but alongside it, a second layer of awareness has emerged, a metacognitive layer, a mind watching the mind. This state is called lucid dreaming, and it is neurologically distinguishable from ordinary REM in ways that have been measured in sleeping people in laboratory conditions. What distinguishes it is the presence of elevated gamma band oscillations in the frontal and frontolateral regions of the brain. Electrical activity in the range of roughly 25 to 40 cycles per second, in regions that during ordinary REM sleep are relatively quiet. A study published in 2014, conducted by Ursula Voss and colleagues, took this observation further. While subjects slept in a laboratory, transcranial alternating current stimulation was applied. A weak, carefully tuned electrical current passed through the scalp and skull into the cortex underneath at frequencies in the 25 to 40 cycles per second range, targeted at frontal regions. Subjects who received the stimulation during REM sleep and who were not ordinarily lucid dreamers reported on waking that something had shifted in the dream. Awareness had appeared. They had found themselves knowing they were inside a dream, watching it, present in it, in a different way. The stimulation at precisely the frequencies associated with frontal gamma activity had, in a meaningful number of cases, tilted ordinary REM dreaming into lucid dreaming. This is evidence not merely that gamma band frontal activity accompanies lucid awareness, but that it may be causally involved in producing it. The gamma activity of lucid dreaming rises against a background of theta oscillations that continue throughout. The broader theta rhythm of REM, the 4 to 8 cycles per second wave that moves through the hippocampus and spreads across the frontal and temporal lobes, does not disappear when lucid awareness arrives. It continues underneath the steady background oscillation of the REM brain, while gamma flickers in the frontal regions above it. Two rhythms coexisting, the slow sweep of theta, ancient and persistent, and the faster brightness of gamma, associated in the waking brain with focused attention and conscious awareness. In the lucid dream, both are present at once. The brain has found a way to be simultaneously in the depths of REM and somehow awake within it, theta carrying the dream and gamma carrying the witness. Verified lucid dreamers in laboratory settings have demonstrated this in a way that bridges the subjective and the measurable. Before sleep, they agree on a signal, a specific sequence of voluntary lateral eye movements performed while dreaming that will appear in the electrooculographic record alongside the rapid involuntary eye movements of REM. When a lucid dream begins, they send the signal. The eyes move in the agreed pattern, left, right, left, right. The record captures it, and the researchers outside the dream know that inside it, the dreamer is conscious. At that moment, the EEG shows its elevated frontal gamma. The EIC reflex is abolished, confirming the body is still in full REM atonia. The brainstem is still generating its waves. And somewhere inside all of that, a mind is both dreaming and watching itself dream, sending a message through the only motor pathway that REM sleep leaves open, the eyes, into a laboratory that is recording it. There is, running through all of REM sleep, a chemical environment unlike anything in waking life. Norepinephrine, the neuromodulator released from the locus coeruleus in the brainstem, a molecule deeply involved in stress responses, in the heightening of attention, in the way the nervous system marks experiences as important and worth retaining, falls to its lowest measurable concentration during REM. The locus coeruleus reduces its firing to near silence. The norepinephrine that during waking saturates the forebrain, modulating how memories are encoded and how emotionally significant events are processed, drains away, and in its absence, the brain enters a chemical state that is found nowhere else in the 24-hour cycle. Matthew Walker and colleagues at the University of California developed a hypothesis about what this norepinephrine reduction does. The hypothesis is called sleep to forget, sleep to remember, a formulation that captures the proposed two-sided process of REM sleep's work on emotional memory. The idea begins with an observation. Emotionally significant memories are processed differently from neutral ones, and much of the difference involves norepinephrine. When a distressing event occurs in waking life, norepinephrine helps tag that memory as important, but it also binds the affective charge to the content, preserving not just what happened, but the full weight of the feeling that accompanied it. The memory carries its emotion the way a physical wound carries its pain. During REM sleep, in the low norepinephrine environment created by the locus coeruleus, going quiet, those memories may be reactivated, replayed, revisited, processed, but without the full effective signal that was present when they formed. The hippocampus and the default mode network move through the emotional material of the day and recent past, but the neuromodulatory context in which they do so is stripped of the chemical that originally stamped the memory with its distress. Over successive REM periods, the hypothesis proposes the emotional charge gradually separates from the informational content. What happened is retained. How it felt becomes lighter. The memory survives. The weight of it, slowly, does not. Evidence supporting this picture comes from several directions. Subjects deprived of REM sleep, but not of NREM sleep, show impaired recognition of subtle emotional expressions in other people's faces the following day. They lose some resolution in reading the emotional signals the human face carries, as though the fine calibration of emotional perception has been disrupted by the absence of the state in which it is normally maintained. The amygdala's responses to emotional stimuli become dysregulated after REM deprivation, suggesting that the processing REM normally accomplishes, the nightly running of emotional experience through the low norepinephrine dream state, is not merely optional, not merely a biological artifact, but a maintenance process on which the waking emotional system depends. The drug prazosin has offered a different kind of evidence. Prazosin blocks the action of norepinephrine at certain receptors in the brain, reducing the effective norepinephrine signal even during waking. It has been used to treat nightmares in people with post-traumatic stress disorder, and in doing so, it reduces the frequency and intensity of the distressing dream content that repeatedly replays traumatic events without resolution. The working hypothesis is that post-traumatic nightmares represent a failure of normal REM processing. The low norepinephrine environment of REM that should allow emotional reprocessing is somehow disrupted in post-traumatic stress, perhaps because norepinephrine levels remain elevated into sleep, preserving the full effective charge of the traumatic memory and causing it to be replayed with its pain intact rather than softened. Prazosin, by pharmacologically reducing norepinephrine signaling, may recreate something of the chemical environment that healthy REM sleep naturally provides. The drug's effectiveness, where it is found, suggests that the chemistry of normal REM sleep, the nightly descent in norepinephrine, is doing something real and necessary to the emotional record. The emotional record is long. Every experience that carries feeling, and there are many more of these than waking life makes obvious, passes eventually through the processing of sleep. The small frustrations of an ordinary afternoon, the unexpected pleasure of a conversation, the slow background anxiety of an unresolved situation, the sharp ache of something lost, all of these move through the dreaming brain over successive nights, each passage through REM sleep, another iteration of the low norepinephrine reprocessing that the brain has evolved to perform. Nothing is resolved in a single night. The process is gradual. Time and sleep together accomplish what neither can achieve alone, and it is possible that what is sometimes described as the natural passage of grief, the way that pain softens without disappearing over months and years, is in part a biological process, the slow work of accumulated REM sleep, gradually separating the memory from its fullest weight. The default mode network during REM is doing something at the intersection of memory and self. When the medial prefrontal cortex, posterior cingulate, hippocampus, and angular gyrus are all active together in the configuration characteristic of REM dreaming, they are not merely retrieving stored experiences, they are combining them in ways that seem to generate new connections between memories that were never linked in waking life. A person or place from one period of life appears alongside an emotion from another. A spatial environment belonging to one set of memories is populated by figures from an entirely different chapter. The dreaming brain does not curate from a stable archive. It draws from across the whole record, simultaneously, without the temporal or categorical boundaries that waking memory retrieval normally respects. This recombination may serve a function that is only beginning to be understood. If emotional memory is to be reprocessed, the reprocessing may be more effective when the memory is encountered in a new context, when it is not simply replayed exactly as it was stored, but encountered alongside other material, connected to other experiences, viewed from angles that waking recall would not provide. The strange associations of the dream, the way a face from childhood appears in a place from last year, the way an emotion felt in one situation colors an entirely different scenario, may not be random noise in the processing, they may be the mechanism itself. The dreaming brain mixing the archive the way a living body mixes compounds, not to disorder them, but to catalyze something that could not happen if each element stayed in its original container. The degree to which the default mode network activates during REM and the coherence of its connectivity during sleep correlates in human imaging studies with the likelihood that the sleeper will, on waking, be able to recall dream content that is emotionally meaningful rather than fragmented. A more connected default mode network during REM produces, on average, a more coherent and emotionally organized dream report. This is not proof of function. The ability to remember a dream is not the same as the dream having accomplished something, but it suggests that the network's coherence during REM reflects something about the quality of the processing occurring, that the more organized the nocturnal activity of this network, the more organized the emotional material it has moved through. A person who has slept a full night and passed through 4 or 5 complete REM periods wakes with something subtly different than they had when they went to bed. The change is not always perceptible. No single night of REM sleep produces a transformation that announces itself clearly, but accumulated over weeks and months, over the thousands of REM periods that constitute a year of ordinary sleep, the effect is real and measurable. The amygdala becomes better calibrated in its responses to emotional stimuli. The recognition of other people's feelings becomes more accurate. The ability to hold distressing memories without being held by them, to recall what happened without being fully returned to the worst of how it felt, gradually improves. These are not dramatic clinical outcomes. They are the ordinary outcomes of ordinary sleep. Small adjustments made every night, the brain quietly maintaining its own emotional competence. The visual cortex runs through imagery that no eye has gathered. The amygdala fires with the intensity of lived experience. The hippocampus moves through memory, combining and recombining. The medial prefrontal cortex, where it is intact, weaves coherence through the activity of the others, holding the narrative together, keeping the self recognizable within the dream, even when the logic of the dream has abandoned every waking rule. And through all of it, in the background, the low norepinephrine chemistry of REM sleep creates the conditions under which feeling and fact can slowly begin to come apart. The pontogeniculo-occipital waves continue rising from the brainstem. Each one travels the same path, up through the thalamus, into the occipital cortex, arriving like a message from below to a visual system that has been waiting. The waves come in clusters, preceding each burst of rapid eye movement beneath the closed lids, and when they arrive, the visual regions respond, and the imagery of the dream deepens or shifts, or a new scene assembles from the materials the brain has to hand. No two REM periods are identical. No two dreams arise from the same configuration of activation and memory and emotional state. Each night of sleep is its own iteration, the same machinery, the same chemistry, the same architecture, producing something that has never existed before and will not exist again. The smallest and most specific things the brain does during REM sleep are also the hardest to observe from outside. The precise moment when an emotionally charged memory shifts, when the amygdala's encoding of that memory loses a little of its intensity, when the hippocampal representation of the event becomes slightly more neutral in its coloring, cannot be measured directly in a sleeping person. What can be measured are the aggregate effects across many nights, the behavioral outcomes of emotional regulation, the subjective reports of people who sleep well and people who do not. But the moment itself, the cellular and synaptic event by which the weight of a memory is quietly adjusted during REM sleep, this remains inside the brain, invisible to current instruments, inferred from the converging evidence of imaging and behavior and pharmacology, rather than directly witnessed. There is something appropriate in that invisibility. The brain during REM sleep is performing its most private work. It has withdrawn from the world, from sensation, from motor output, from the social and practical demands that occupy it during waking hours. And what it does with that withdrawal is process the residue of experience in a way that serves the days to come. The amygdala's high activity is not a malfunction, not a warning signal, not an artifact. It is the emotional brain at work, doing in darkness what cannot be done in light, touching the material of experience that the press of waking life does not allow time to examine. Every night this happens, not occasionally, not in moments of particular stress or processing need, but every night, across every life, from very early in development onward. The dreaming state is not a luxury or a complication of the brain's organization. It is a scheduled maintenance of the emotional system, recurring with the reliability of the body's other rhythms, the heartbeat, the breath, the turning of the sleep cycle itself. The brain that will face another day needs to have spent some hours at this work first, not consciously, not deliberately, not with any awareness of what it is doing or why. The process runs without instruction. It has always run. It runs now in the millions of sleeping brains that are somewhere in REM sleep at any moment of the night, each one working through its own record, each one moving toward morning with its emotional register quietly, incrementally adjusted. The amygdala still burning, the visual cortex generating its interior world from the brainstem's rising waves, the hippocampus turning through its archive, the default mode network holding the dreaming self in its distributed embrace. And in the chemical quiet of the low norepinephrine night, the long, slow work of separating memory from its weight continues, unhurried, self-contained, asking nothing from the waking world. This is what the sleeping brain does with the dark. Not waste it, not merely sustain itself through it, but use it, use the hours of stillness and withdrawal to perform something that the noise and light and motion of the day do not permit. The dream is the surface evidence of this process, the part that occasionally breaks through into memory on waking, fragmentary and strange and already fading. Beneath it, the real work is chemical and electrical and synaptic, invisible and continuous, older than language and more fundamental than anything the waking mind deliberately chooses to do. The feeling of a painful memory growing lighter across the months, the face of someone long absent appearing in a dream and carrying less grief than the last time, the slow, almost imperceptible softening of what time and sleep together have touched. This is not mystery, or not only mystery. It is the brain doing what it was built to do, night after night, in the low norepinephrine quiet behind the closed eyes, in the warmth of the amygdala's patient work, in the default mode network's long unhurried movement through the material of a life. The last REM period of a full night stretches longest. By the fourth or fifth cycle of sleep, the REM episode can extend to nearly an hour, a long window of the brain's most interior activity, arriving in the hours just before the ordinary world reasserts itself. The body has been still through all of it. The muscles have been held in their glycinergic silence. The brainstem has continued generating its waves. And the emotional record has been moving through the low norepinephrine state of the dream, not solved, not erased, but touched, turned over, recontextualized in the strange associative logic that only the dreaming brain employs. When the last REM period ends and the body begins its slow ascent back through the lighter stages of sleep toward waking, something has happened that did not need to be witnessed to be real. The chemistry has shifted. The synapses have been adjusted at a resolution too fine to feel directly. The amygdala's calibration has moved a small increment in the direction of equilibrium. Nothing dramatic, nothing sudden, nothing that the morning will clearly announce.